Welcome to my course Industrial Biotechnology. Why the heat is considered as the effective way for sterilizing the media, and we have shown you that how temperature plays very important role for killing the microorganisms, and we have shown that as you increase the temperature, your sterilization time drastically reduce. And also I try to discuss that why in the industry the batch sterilization is not preferable as compared to continuous sterilization process, because batch sterilization process not only required the higher amount of steam, but also time of sterilization also about double as compared to the continuous sterilization process. Now, this class I am going to share with you that continuous sterilization process, how that is in practice in the industry. Now, if you look at the continuous sterilization process, we have direct steam injection sterilization, we have plate heat exchanger sterilization. Now, let me explain this process, because direct steam injection means you mixing the steam with the media. Now you can see it here that raw media is coming one way and steam is coming in other way, and this is the actually we call it holding section. You hold this is the pipeline, and this pipeline will be insulated. That I want to draw it like this, and this pipeline is basically they're insulated. That means it is the insulated like this, so that the heat loss, this is the insulation that we have with the outside the pipeline, so that heat loss should be minimum. So that is in practice, because we pass media here, and now if you know the length of the pipe, suppose length of the pipe is about L, and if you know the velocity of the liquid, what is velocity is u. What is the unit of velocity is centimeter per second, am I right? And what is the unit of length? It is the centimeter. Now the time, the holding time, the liquid resides in the holding section will be what? This is L by U. So you divide by, so you can easily find out. Suppose you want to have 2 to 3 minutes holding time, so you can adjust accordingly. This is like this, and then there is the expansion valve, and through this expansion is expand in the particular in a chamber, what you call it flash cooler. Then what happens that from the top vapor will be released, and concentrated liquid will be falling like this. Now here there is a problem that if you do this, that there will be loss of moisture that will take place. Another most important thing is that when you mix the steam with the raw media, the steam undergo condensation. Am I right? In the particular pipeline, and since the condensation of the steam occurs, then the viscosity and density of the liquid will be changed. Then, as you know, the flow characteristics of the fluid depends on largely depends on the viscosity and the density. I can give the example of the Reynolds number. Reynolds number is equal to what du rho by mu. So with the density and mu, that plays very important role. Now, as your density and the viscosity changes, it is very difficult to maintain a particular temperature inside this holding section. And if you are unable to maintain a particular temperature, then your sterility it's very difficult to maintain. So that is the major problem that we have this direct steam injection process, and that is why the industry this is not in practice. Now here is the temperature time profile, the temperature profile of this system. You can see that as soon as the media injected, the instantaneously temperature rises, because that is the major, there is no heat loss basically, that your whole stream is directly, the heat is directly transferred to the media. So that instantaneously temperature will rise to 140 degrees centigrade and you can hold it. Then you can do the flash cooling like this, 80 degrees centigrade, then slowly you can cool it down to 37 degrees centigrade. This is what we have, but this is not in practice in the industry. And industry, the sterilization technique that is in operation, that is we call plate heat exchanger sterilization. Now here we have 3 heat exchanger, 1, 2, 3. You can see the 3 different heat exchanger. Now this particular heat exchanger we call it economizer, and this is this we consider as the economizer. The reason is that I can explain that the raw media is going like this, and it is coming out like this, again going to this is the steam injection, and after going through the holding section it is. So what I wanted to tell, that here you raise the temperature to 140 degrees centigrade, and then you are keeping in the holding section, you cool it down, and for this steam you are passing through the heat exchanger, and you are utilizing your raw media here just to use this heat for preheating the media. So you save some energy here. That is, we call it economizer. This is called you save some kind of heat we here, and then your temperature will come around to say 140 to 80 degrees centigrade. Then we pass through the chiller to have, this is you pass the cooling water. Now then we get the sterilizing media. Now let me explain what is called how the plate heat exchanger looks. Plate heat exchanger looks like this. So basically it is a plate like this. So several plates, they are bunching together like this. So we have this different plates, they are bunching together. Now this surface is corrugated. The surface is corrugated. Why it is corrugated? So if you make the surface corrugated, then what will happen? The surface area will increase. We know the rate of mass transfer depends on the surface area. More surface area, more will be mass transfer. Now what we do, suppose you are passing your media here. Suppose your media here, and you are passing your hot heat maybe steam you are passing through this, or hot media you passing into this. So when you pass this, this plate will come in contact with each other, and then it will heat your desired media and take it out from this particular the sterilizer. So all this plate heat exchanger, 3 plate heat exchanger, this is like this. Similar, this is parallel. The plates are they are bunched together. So several plates, maybe 50, 100 plates, they are bunched together. Now problem is that this particular sterilizer has severe problem. What is the problem? That these plates, one place when binds with each other, you put a gasket here. In between this, we put a gasket. Then we can tie it together. And since suppose we are using the steam as very high pressure, there is every possibility that when you pass the steam at a high pressure, the leakage of your gasket may burst. And that if the gasket bursts, then what is there is some steam leaking is there. Suppose you in between when you pass your liquid, if some leakage in the gasket is there, some kind of cracking in the gasket is there, then also your media can goes out. Those kind of problem that we have. Another problem is that suppose this is the steam, that means it is a high temperature and this is the liquid. So there is a possibility that the surface will be there is scale formation on the surface of the plate. Now if there is a scale formation on the surface of the plate, then what will happen? The conductivity of the material will be lost. If the conductivity of the material lost, then what will happen? More steam will be required, more heat will be required for heating the media. So that is not desirable. So what you have to do, you have to open this plate and clean the plate time to time, so that you can have the effective heating and cooling in the system. So this is the major drawback of this particular process, but this is largely in operation with the industries. Now here I have given you the temperature profile. Here you see that when your raw media is coming, it requires some time to heat the partially heating, and then it is heating to 140 degrees centigrade. This is the, so you require some time, about 20 seconds is required for heating purpose, then 2 to 3 minutes for holding, maintaining the temperature 140 degrees centigrade, or here it is 144 degrees they have mentioned. Then this is 20 seconds for cooling purpose. This goes here and then pass through the chiller. The whole thing that required holding, the time required for that is 20 seconds. So this is how the sterilization is done, medium sterilization done in the industry. So let me discuss some problem related with the medium sterilization process, so that we can have better understanding on the process. Now the problem is like this: media as a flow rate 2 meter cube per hour is to be sterilized by heat exchanger with steam in a continuous sterilizer. The heat contains bacterial spore as a concentration of 5 into 10 to the power 12 per cubic meter. The activation energy and Arrhenius constant for thermal destruction of these contaminants is 283 kilojoule per gram moles and 5.7 into 10 to the power 36 our inverse respectively. A contamination risk of one organism surviving in 60 days operation is considered to be acceptable. Now the sterilizer pipe has an inner diameter 0.1 meter. The length of the holding section is 24 meter. The density of the media is 1000 kg per cubic meter. The viscosity is 3.6 kg per meter per hour. What is the sterilization temperature required? So this is I hope you understand this problem, because what I suggest that we should have a understanding on this process, that how it actually in practice. So you can write a sterilizer like this, and where they have mentioned that the inner the diameter of the pipe, this 1 meter. This is the inner diameter of the pipe. So ID, what I call inner diameter, is considered as ID. Now the flow rate of the media is about say about 2 meter cubed per hour, that is given, and media contains about number of spores is how much? Number of spores, bacterial spores is about 5 into 10 to the power 12 per cubic meter. Am I right? Now activation energy that is given. The contamination, this is very important. A contamination risk of one organism surviving every 60 days operation is considered. Now either in a continuous flow reactor, then first you have to calculate how many organisms is present if you operate this for 60 days. So what we can have, this is 2 meter cube per hour is the flow rate, am I right? And then how much is the organism presence? 5 into 10 to the power 12 per meter cube. So this is, that means how much organism per hour is going. This is, you multiply it by this factor. Now you can convert it to per day, am I right? If you multiply by 24, 1 day is equivalent to 24 hours. So we can multiply by 20 days, you can per day how much organisms are entering into the system. So he is saying that 60 days operation. So you multiply it by 60 days. So how much organism will be there? 1.44 into 10 to the power 16. This is the number of organism. Now he is saying that one organism out of the 60 days operation is acceptable. That means I can write N2 by N1 value 1 by this, and this is equal to 6.9 into 10 to the power minus 17, that we can write. Now from the flow rate, because we know the inner diameter this of the pipeline is 0.1 meter, and this what is the cross section of this? This is pi r square. Pi r square is the cross sectional area. Now if you divide by flow rate, divide by cross sectional area, suppose flow rate is the volume by cross sectional area, what you will get? You will get volume per unit time, and by cross sectional area you will get that you will get here you will get meter per hour. That means length per hour. You will get some length per hour. That is exactly how that is meter cubed, and this is meter square. So this will be meter per hour. So velocity you can easily find out. Why we want to find out velocity? Because we want to have the Reynolds number. Reynolds number is du rho by mu. So if you know the diameter of the tube is 0.1 meter, and velocity is 250.4.6 that meter per hour, and the rho is 1000 kg per rho of the water is like this per meter cubed, and viscosity is 3.6 kg per meter per hour. Then this Reynolds number is coming about 707 into 10 to the power 3. Now the Piclet number, we can from this we can calculate the Piclet number. The Piclet number, how we can calculate? We can calculate the Piclet number equation is that U into L by dz. Dz is the that dz value we can calculate from this equation, the dispersion coefficient, that we can calculate with this. We have a plot between the Reynolds number and this dispersion coefficient here, that this is diffusivity, this is the velocity, this is the kind of thing. So if you know the Reynolds number is corresponding value, you can find it out. And if you know that from this Reynolds number is 7.07 into 10 to the power 3, we can determine the dz value. That either experimental, let us choose the experiment. So this is from this figure. We can have this from this figure. We can have this value there somewhere here, and then we find out the z value, which is coming about 16.6 square meter per hour. Then your Piclet number is equal to UL by dz. U is the velocity, that is like this, to 254.6 meter per hour into 24 meter is the length of the tube. Then divide by dz value, you will get 364.8. Now figure, if you look at the figure 2, it is the plot between the N2 by N1 against the, this is the what you call damp Polar number, KDL by U. So you have different values here. So you can find out that if you know this value, corresponding value you can find it out. So you can if you n by this value, N2 by N1 value is 6.9 into 10 to the power minus 17, and Piclet number is this. Then DA value is coming about 42. If it is 42, and actually that if you look at, this is the different Piclet number. Piclet number 10, 20, 30, 40, 50, 70 like this. As your Piclet number changes, this will be becoming steeper and steeper. So we can find out this value for 42. Then KD equal to what is this equation equal to U. If you look in this equation, dA equal to KD. KD is the thermal death rate constant, L by mu. So if you know the dA value and you know L value, you know U value, so you can easily find out the value of KD. KD is coming about for 445 into 0.6 into our inverse. Now once you get that, now your situation is little bit simpler, because we know as per Arrhenius equation, this equation is there, the Kd equal to A into e to the power E minus Ea by RT. Ea, this will be Ea by RT is the activation energy, the energy required for deactivation of the cell. Ed also sometimes is right. So we can do this and find out what is the temperature required for the sterilization. So all these values is given here. Your activate the activation energy required for killing the organism, death of the cells is given, Arrhenius constant is given, gas constant we know. Then you put this value, we will find this temperature. So the temperature of the sterilization is coming about 125 degrees centigrade. So this is how we can calculate the temperature of the medium sterilization, that we can easily calculate. Now that let me show you how we can transfer the because now we have the information with us how media can be sterilized, how air can be sterilized. Now things that remaining with us, that the culture we prepare in the lab, and that is to be aseptically transferred in the inoculum vessel, because I told you the volume of the inoculum vessel usually 5 to 10% of the production fermented, and I work with industry where the capacity of the production fermenter was 200 cubic meter, and inoculum vessel, that is where we prepared the inoculum, is about 5 to 10%. In our case it was 14 cubic meter. So 14 cubic meters means 14,000 liters. The 14,000 liters culture we cannot prepare in the lab. So that is why you have to prepare this inoculum in the fermentation plant itself. So what we do in the lab, what we prepared the seed culture. We prepared the seed culture, and through our research and developmental work, we try to we determined that what is the optimum concentration of cell required for the optimum amount of required for the preparation of the media, that we shall have to determine. And once we have this value, I can give a very typical example, that in case of bacterial fermentation process, particularly for the unicellular cells, we don't have any problem, because the unicellular cell you can count the number under the microscope, and you have hemocytometer, so you can count the number, what is the concentration of cells. And even you know, if you know the mass, the total mass of the cells, and if you know what is the weight of individual cells, from that also you can convert it, that how many cells are present there. It is not very difficult. Either you can take the mass, or you know the density of the cells, you can easily find out how many cells are there. So now question that is we face in case of the fungi. Fungi is a multicellular organism, it is a filamentous growth. So there it is not a unicellular cell. So how you can count the numbers? So in case of fungi, we use the spores, because the under stress condition it produces spores, and spores can be counted, because we can count the spores. We can under the hemocytometer we can count so you have to quantify that how many spores is required. So in case of unicellular cell, we don't have any problem. We can use it directly unicellular cell or the yeast cells. We don't have any problem. We can count the number, and we can find out that what exact number of cells is required, what number of cells exactly required for the preparation of the inoculum. So that number you have to maintain. Then so that we put it in the seed can like this. Seed can looks like this. What is the seed can? It is here we have a valve, and here we have a valve. Now here we have some nut and bolt arrangement. Now in the industry, what you do, suppose this is the inoculum vessel. So if you want to transfer this, here also we have valve. So here when you bring this seed culture here, so we can here we have valve, here we have nut and bolt arrangement. Here also we can club together, and here also we have valve, and here we have nut and bolt, and here we have another pipeline. Maybe it is connected with sterilizer. So it is like this, and also there is another line. What you have, steam line, that is for sterilization of the that line, pipeline. So you have to sterilize this pipeline before you transfer. So you have to pass the line, so and take it out to ensure that line is 100% sterilized, and then you have to sterilize this line also, and then you take it out. The stream you can take it out, and when you ensure that your pipeline is 100% sterilized, then you close this valve. Then you open this valve. Here there is valve is there, so there is nothing is there. So if you open this valve, the air will go here, and here is closed, and you open this valve, this is closed. Then with this air pressure, whatever seed culture is there, that will come to the inoculum vessel. This is IV. You can transfer it, and when it is completed, you close it. Again you pass the steam here, so that whole pipeline will be sterilized, and then you run this operation. But when you get the culture, now how you do that? How you transfer this culture from IV, that inoculum vessel, to production fermenter. Suppose this is a small vessel, small inoculum vessel. This is inoculum vessel that we have, and this is big production fermenter. This is IV, and this is production fermenter. So here also you have like this. So what do you do? That here we do sparging. This is air, am I right? Air in, and this is air out. Now if you close this air, then what will have? There will be tremendous pressure here, air pressure. With that air pressure, liquid will come here, and you can fall it here. So you can transfer the culture easily aseptically. You can have the valve here. You open this valve and drag the culture from this vessel to this vessel. So this is how we can, and then when it is transferred is taking place, then we can close this valve. You close this valve, and again we do the sterilization of the valve. This is how we can aseptically transfer the media, transfer that culture from the inoculum vessel to the production fermenter. So this is in practice in the industry, and this is found very effective. Thank you very much.